Hello everyone, welcome to the demo of our Immunology Lab activity. And what this is going to be today is a simulation of blood typing assays in which we're going to see if blood that was found at a crime scene matches the victim of the crime and any of the four suspects. When you guys watch this video and look at the results, you're going to be checking for an agglutination reaction in the blood typing results, and you're going to interpret what that means. You're going to determine the blood type of each sample, and also if there's a match between the crime scene blood and any of the suspects. In addition to that, you're going to look to see if the crime scene blood matches the victim's sample. So there are a few terms I would like to introduce you to today before we get started with the activity. Firstly is the term antigen, which an antigen is a substance that stimulates an immune response. It's usually going to be a protein or a polysaccharide. And then a B cell is a type of lymphocyte, which is going to be a cell in the immune system that's capable of secreting an antibody protein. And what will happen is that when it encounters a suitable antigen, an antigen that it recognizes, then it will secrete an antibody that binds to that antigen. Now, an antibody, of course, then is a protein produced by B cells in response to an antigen. And today, we're going to be looking at the reaction agglutination, which is the process of when an antibody binds to its antigen and causes cross-linking from antigenic particle to antigenic particle that results in clumping. And this is a reaction that is going to be visible today. And also, just so that you know, we're technically looking at hemagglutination, which is agglutination of red blood cells specifically. So the idea, like you can see down at the bottom of this slide, is that if we have type A red blood cells that encounter anti-A antibodies, which are antibodies that are specific to bind to type A antigens, what will take place is that cross-linking will occur between two different red blood cells of that type A blood type due to the presence of the anti-A antibody, which will result in agglutination of these type A red blood cells. Now, the ABO blood typing system that we hear about in medicine, and many of you probably know your blood type, involves type A blood, type B blood, type AB blood, and type O blood. In addition to that, like let's say you have AB positive blood, what the positive refers to is the presence of an Rh factor on your red blood cells. So if you are Rh positive type AB, then you would be called AB positive. If you're Rh negative type AB, then your blood type would be AB negative. Now, this is with the understanding that the immune system, or your immune system, is tolerant of your RBC antigens. But if other RBCs that contain other antigens are introduced to your blood, there's the potential for agglutination to take place, depending on the blood type that you have and the types of antibodies that you have present in your blood. So if we look at this chart here, type A blood types will have anti-B antibodies in their plasma. On the outside of their RBCs, we would find A antigens. And in the case of an emergency, people with type A blood can receive blood from either type A or type O donors. And the significance of that is that in an emergency situation, a patient whose blood type is not known can actually receive O negative blood on a temporary basis until blood typing can take place to deal with the immediate acute needs of the patient. Now, with type B blood, these individuals will have anti-A antibodies in their plasma. The outside of their red blood cells will contain B antigens, and they would be able to receive blood from individuals with type B blood or individuals with type O blood. Now, for someone with type AB blood, there are not going to be antibodies for B or for A in their plasma. So they will not have anti-B antibodies or anti-A antibodies. The reason for this is that their red blood cells actually contain antigens that are both type A and type B. Now, AB positive is considered the universal recipient in emergencies because they can receive blood from type A, type B, type AB, or type O. The reason why AB positive is a universal recipient and not AB negative is because that AB negative would actually have Rh antibodies in their blood. Now, someone with type O blood has both anti-A antibodies and anti-B antibodies in their plasma, and their red blood cells do not contain A antigen or B antigen. Now, O negative is considered the universal donor because those red blood cells do not contain A antigen, B antigen, or Rh antigen. So just as a quick reminder of what we're doing, we have 4 blood samples from suspects. We have 1 blood sample that was found at the crime scene. We have a blood sample for the victim. And then we also have anti-A, anti-B, and anti-Rh antibodies. We're going to be doing the tests on these spot plates like the one pictured here. So we're going to start by testing the victim's blood. So we're going to put 1 drop of the victim's blood in each of the 3 wells on 1 spot plate. We are going to put anti-A serum or serum containing anti-A antibodies into the well labeled A. And what this will do is if there are antigens on the RBCs that are type A, that sample in that well will agglutinate. Then we're going to take anti-B serum or serum containing antibodies that will bind B antigens and put a drop of that in the well labeled B. And once again, a positive test will indicate that there are B antigens present. And additionally, we're going to use anti-Rh serum, which contains antibodies that bind Rh antigens. And a positive test in that well will indicate that the sample is Rh positive. Then we want to mix all 3 of those wells, being sure to use a clean toothpick for each one so we don't cross-contaminate. And then we are going to wait several minutes for that sample to develop, for that sample to have a chance to agglutinate, and we're going to move on to the next sample. Now we're going to test suspect number 1's sample. Since you've already watched me go through this process 1 time, we're going to go ahead and speed it up a little bit. And now we're going to move on to suspect number 2. And now time for suspect number 3. And it's number 4's turn. And now it's time to do the suspect blood that was found at the crime scene. So this is the sample that's going to be matched against the 4 suspect samples to see if one of them may have been the one responsible for the crime. And then it will also be checked against the victim sample. And then, as a reminder, the A well on the spot plate is going to indicate whether or not A antigen is present on the red blood cells. The B well will indicate whether or not B antigen is present, and the RH well will indicate whether or not the RH antigen is present. And the results will be visible in a moment. What you're going to want to do is pause the video as each of the individual slides comes forward on the screen so you can determine in which wells agglutination took place so that you can determine the blood type of each sample. You can use page 70 of your lab manual to record your results, and then there will be some questions in the worksheet assignment about the different blood types and who might have been responsible for the crime.